Hello?
Doing well.
Hello? There we go. Unmute. Okay, so slightly earlier than expected. I'm Penn, and so first out of these large number of authors, and I'm going to be talking to you about our paper parameterizing a computational model of supercoiling in plasmid DNA with a biophysics model of supercoiling in plasmid DNA. Wouldn't all fit, so we cut it a bit short. So this is basically us doing the drawing on the back challenge of modeling. So when we're doing modeling of biology, obviously it's really nice if we can look at some nice tidy biology and just model it. But the reality is these things are really small and really complicated, and people keep looking at different forms of them. And so we can't just look at it once and go, yes, that's what it's doing, let's go model it. So instead, we're doing a lot of very complicated stuff to produce very complicated models. And then we've also got, we've got very big, very complicated models, which aren't very good if you want to do things like machine learning, or you want to look at lots and lots of different models of something. So we're trying to also make some smaller, easier to use models that just look at, say, the computation. But first, what are we actually modeling? So we're modeling supercoiling driven circuits in plasmid DNA. And I'm pretty sure some people in here don't know what that is. So let's start with what supercoiling is. So DNA is a double helix, and if you have it in a relaxed state, it's got 10.5 turns per base pairs. So you've got all these base pairs, ATCG, along the DNA, and for every 10 of those pairs, it does 10.5 turns. On the bottom here, we have what happens when you start doing stuff to the DNA. So when you want to copy a bit of the DNA, you have to go in and grab it, open it up, connect to it, and then you have to move along and you open up the DNA ahead and you close the DNA behind. But what this does to the twist of the DNA, because it's a physical thing, is that one side gets overwound and the other side gets underwound because the oils can't escape. So we start with a plasmid, a circle of DNA, and what we want to do is turn on a gene in the DNA by opening up part of the plasmid by changing the coiling. So basically, and if we're doing this in the biology, we turn on something that produces a fluorescent protein that you can see inside. See it on a plate reader or something.
We just have, they're not visible on the Zoom.
Oh, okay. Yes, because Lana would have been shit. That's fun. So then we also have our abstraction chain. So on the biological end, we start with, on the biological end, we've got an actual plasmid, something you build in the lab and can then observe with lots of fancy equipment that can look at certain things, but not other things, because it's all very small and very hard to see. Then we have a physical model. So our physical model is based on all the biological experiments that have come before that our physics person has read and found in the massive literature. So it's based on many observances of an actual system, but it is not, but it is an abstraction of some sort. So in our case, we've got a line of DNA and we've connected some things to it, and they come on and off and we see what they do to the supercoiling as we go. Then we've got the computational model, well, our small, quick model that just goes, okay, so based on what the physics model said, and based on what we observed from the biological model, if I do this, this gene turns on, on and it produces this thing and it turns off. And in our case, we've got a bit like, you may have heard of gene regulatory networks before, but in our case, we've also got, and what do they do to the supercoiling and what does that do to the gene regulation? If you're interested in the details of the model, there was a paper from last year here that's got all of that, including some wonderful maths. Apparently it's technical difficulties day.
That's because you just frozen on it.
Okay, so we have two main components in the system we're modeling today, and one is TET-A, which is TET-A is just the name of the gene. It produces something which I wouldn't want to spell and I'm not going to attempt to pronounce. But what it also does is it creates a very large amount of negative supercoiling, so that's the unwinding, as it copies. And it does that just upstream behind it. So if it's pointing left, the negative supercoiling goes right. Or clockwise and anti-clockwise, as I should say, because we're on a circle. We also have a promoter. So a promoter goes in front of a gene, and you have to kind of switch on the promoter in order to copy the gene. In many systems, this is done with a chemical signal or a transcription factor. But in this case, we've got a promoter designed to turn on when it gets very negatively supercoiled. All nice and simple so far. Thing that produces large amounts of negative supercoiling, thing that turns on based on large amounts of negative supercoiling. And we put this into a plasmid, so we've got TET-A driving our supercoiling. We've got our promoter. We've got some other stuff that's mostly there, so this actually copies and can go into bacteria, because we do actually put this in bacteria, which is fun. And we've got N-calma here, which is a gene that produces blue fluorescent protein, that you can see in the lab under the plate reader if it turns on. So so that's the design of this plasmid. So what do we need in terms of parameters for our computational model? So first, we need to know how much negative supercoiling does TET-A actually produce? Because that's going to be pretty important since this is the engine driving our system. How much negative supercoiling does our fluorescent gene produce? Because like I said, anytime you copy a gene, you're moving this thing along that coiled helix, and it produces positive and negative supercoiling. You'll note I'm ignoring the positive supercoiling from TET-A because we've done some funny stuff that means we can ignore that. Don't worry about it. It's there, but we can ignore it. So how much is that producing? Because it might be interfering with things. Then we have two more. Well, that's not something funny, but anyway, so this is supposed to be a sigmoid curve on the left, but apparently moving this presentation between things has changed the graphics slightly, so it now isn't quite so sigmoidal, but oh well, we carry on. So we've got two different possible things here, which are the response curves for our promoter. So when does the promoter turn on and by how much? Because it's not actually a switch, it's not an on-off. We want it to respond as it does in biology in a somewhat gradual and then sometimes less gradual way. So we ended up with two possible forms of this. So there's the sigmoid and there's the binomial curve. So we've got for each of them a response point. For the sigmoid, that's in the middle. So when is it most of the way towards being on rather than off? And for the normal, the response point is when is it most on? And then both of them have a gradient as well, so we can say how sharp these actions are in our biology. Finally, we have the output. How much protein do we actually produce? Well, not protein already, but close enough. How much do we actually produce from this gene? Because that's going to be important when we want to do comparisons. Finally, supercoiling doesn't stay there forever. If we just kept producing all this supercoiling and it never went anywhere, these would explode. I'm not joking, actually explode. They can actually tear themselves apart with supercoiling if you let them. So there's some cool enzymes and stuff in the cells that come and fix all of this. But what we want to know is between time steps, how much do of what we did this time step goes away. How much sits around in between? Okay, so that's our supercoiling relaxation. So that's what we need for our computational model. This is the model we've got to get it from. So this is TalkViz. This has been put together by our fantastic collaborator, Victor, who has this wonderful model. It's got the DNA, and we've got a series, we've got genes, which is the blue arrow. We've got NAP sites. Don't worry what that stands for, I can't remember right now, but it basically means things that aren't genes. Genes, but where stuff connects to the DNA. Okay. And you won't be able to see those, but they're under the green blocks. So that's your DNA, and then you've got effectors. So you've got things like the red cloud that connect to the DNA and add positive supercoils. You've got things like the blue cloud that take them away. Those are those fun enzymes that stop everything from exploding. Then we've got our blocks, our green cubes. These are things that stop supercoiling moving around. So if enough stuff grabs onto a bit of DNA, or if the DNA forms a little knot somewhere, then the supercoiling can't get past the knot, can't get past the big thing connected to the DNA. Much like if you're twisting a rope and someone just grabs part of it and you keep twisting, the twisting doesn't go through your hand. And then finally, we've got the white circles, which is the actual copying mechanism. So this is, oh, I always get this wrong, RNA polymerase. That's right, excellent. As it moves along the DNA, copying the gene. And this is all done by all of these things come along, they bind to the DNA, they have an effect, and then they unbind. And the effect can be on the supercoiling predominantly, because that's what we're modeling here, but it can also be on things like how many copies are created of stuff. So that's the model. Here's what I actually take from the model, or here's what I can get out of that model. So I get the binding rate. I have the number of things bound to a gene at any particular time step. How many of these RNA polymerases are attached to a gene right now? And one of the things I was able to calculate from this is a full transcription takes 22 time steps. And we'll come back to that number in a minute. The other thing we have is supercoiling density. So we mentioned promoters. Promoter is the bit that's got to open for the gene to copy. Okay. And we can find out what is the supercoiling value there, how many twists are there per 10 base pairs, is at that point when the transcription starts, when we first bind to this gene and start copying it. And since the promoter is supposed to be responding to negative supercoiling, we're kind of hoping that that number is going to tell us how much negative supercoiling it takes to open our promoter. So how are we going to compare these two models once we've got all this? So I'm going to go through this pretty briefly. The maths is all in the paper, but basically we have to compress time a little because the physics model is very, very detailed, and there's like 5 steps in the physics model to 1 step in the computational model. And there's also 95 runs of the physics model that we want to average out for parameterizing our computational model. So that's where those numbers are coming from. But we're going to take the number of effectors bound at each time step. We're going to divide it by 22 once we've done all the summing and math and averaging. And that's going to tell us, in short, how many transcriptions have happened in that time step in terms of the computational model. And then in the computational model, since we don't need to do any time compression and we don't need to average anything, we just go, how much stuff did the computational model produce last time, have in its bucket last time step, how much has it got in this time step, take one away from the other, that's how much it produced in the last time step. And we'll take the absolute difference of that over each point in time and we'll divide it by 1,000. So on average, how different is the stuff being produced by these models throughout the whole run? So we've got a measure of how good is our match. We've got a load of parameters to set. Random search time. We considered a grid search, but then was like, why take a sampling of even things around the place when we could actually get really good coverage of everything by picking them at random? And we did get good coverage. There's a graph for that next. But we also had to set reasonably realistic numbers. So if there's any biologists or people who work with any of this in the audience, yes, we have limited it so it doesn't, or so it can't get to really explodey amounts of supercoiling. But we have also, but on the other hand, I have allowed it to possibly do a transcription every 5 time steps of the other one, which we know take 22. So it can, in theory, if it really wanted to, go ridiculously fast. But hopefully it's going to tell us it doesn't want to do that. So we ran a random search and we got this nice graph out, and on the diagonal you can see how much of things in each bucket we selected in the random search, so we got fairly good coverage of everything. On the horizontal and vertical axes is each of our parameters. And what we were hoping we'd see is some trends. We were hoping to see we got better M values for certain ratios of parameters. We didn't. So I went through and I looked at all of the parameters individually to see if there were any trends there. And this was the best I could find. Turns out this model is very insensitive to basically everything. But we do get a slight lower is better for our max output for M calima. So I went and I grabbed the best value here. We'll come back to it in a minute, but we tried some stuff with, what if we fix that value and then look at the other parameters? Maybe if we fix some of these parameters, so there's less variation overall, we'll start to see some signal. But before we look at that, we also went, okay, so random search didn't work. What about drawing directly from what we can see in the model? So we said we could see supercoiling locally. And this is the supercoiling report at the promotion. I took one look at this and went, that is not a sigmoid. Right. So this is where the other curve came in. And so we tried, what if we ran this again, but we allowed it to be the binomial curve instead? Would we get better parameter setting from that? And the results were mixed. So fixed max, it turns out, has a very large effect on all cases. If you fix the max output, it's very happy with that. It likes that a lot. That always produces a large effect and a significant improvement, whether you're using the normal activation curve or the old sigmoid. The normal activation curve improves your best fitness. At least on its own. With the fixed max, it didn't quite improve our best fitness, but it got us very close to the same fitness as the fixed max. It also, however, widens your distribution and has no actual significant effect on the distribution. I, however, am suspicious of this fixed max value on its own with the sigmoid. I am highly suspicious of the fact that this is so good, because I think it's just turning it on and producing at a rate that's close enough to the physics model to cheat the system, as it were. I actually much prefer the normal fixed max, because there it's actually having to moderate its supercoiling. It can't just accumulate huge amounts of supercoiling enough to turn itself on and not care about what happens afterwards. It's because if it overshoots, it's going to turn itself off again. So currently, that's the one we're more interested in pursuing in future. So in conclusion, we can fix some of our stuff. So we can work out our promoted response curve. We can work out M calima max bit. But the supercoiling, how much is one producing? Are any of these producing? How much is relaxing between times? We can't even find ratios for that. And the simple answer for that is partly because the numbers in the physics model are also made up. Because you can't measure local supercoiling in the lab. In order to measure these things, you take them out of the bacteria and look at them under other stuff. You put them through gels, which means these numbers are very hard to get at. And we certainly can't see them as it's happening. So these little bits of supercoiling that are happening as transcription happens and being generated in that way are extremely hard for us to see in nature. So what we actually need is a lot more experiments where we can look at the results of the supercoiling to try and infer what's going on dynamically in the live model. And for that, we need Alap. Alap is our other collaborator. He's the guy in our lab who is doing a fantastic job producing oh so many plasmids in oh so much bacteria. And we're hopeful that soon we'll be able to have enough data from that to start hopefully parameterizing the physics model better, and then that'll lead into better parameterization for the computing model. But for now, we're a little bit stuck because, as I say, these things are hard to look at, and We could use more stuff from other people's experiments. Other people's experiments are different. And if we go back to that twisted rope, it really matters the distances between the ends of the rope and the bits that's twisting. That's going to change a lot. It matters which way things are moving along the rope. This is a physical system. The context matters. So while we can use other people's experiments, they're not particularly going to be more reliable than what we have now. So thanks to the whole talk team who have been great with this, and our funders, SFI and UKRI's EPSRC. Thank you very much.